as we go forward, right? Yeah. Sorry about that. When I lecture in the fall term, it's in Milam Auditorium, and that room has got the worst flipping sound system I've ever seen in my life. And depending on where I walk in the room, as you guys see, I'm a walker. The wireless microphone gives feedback to this thing, and it's just dreadful, you know. And I sometimes have to just turn the damn thing off and shout in the room, so because people are getting headaches, people are getting all kinds of complaints, and it's just terrible with that. So. Sorry about that. You should have said something sooner. I would have done something about it. I honestly didn't hear it. So, there's a, what is it? There's a, how does this go? That there's a, I forget how this goes. There's a, of course, there's a sound that young ears can hear that, that older ears can't hear. The older you get, the less you get the high, the mosquito ringtone, right? And the mosquito ringtone is used by students to bypass that. But there's also a way for professors to actually buzz their students, and doesn't affect the professor at all. So, so I guess it goes both ways. Okay, let's see. Let me make sure, since I got interrupted here, let me make sure I've got this camera running because I don't want to lose that video, and then we'll be set. We are running, okay. Okay, so, excuse me, I'm sorry. All right, so I've given you now an introduction into mechanisms of allosterism. And allosterism is a very common control mechanism that cells use. In fact, most metabolic pathways, not all, but most metabolic pathways have at least some enzyme that responds allosterically to the environment to control a major enzyme. Feedback inhibition, which you see here, is the most common allosteric mechanism, but it's not absolute, all right? Meaning that it's not always the very first enzyme in the pathway that is the one that is controlled. We'll see when we talk about glycolysis at the very end of the term that glycolysis is very odd in that it's regulated at 3 different places. And there's a reason why, but it's not regulated by feedback inhibition. Okay. All right. Why your book talks about these enzymes here, I have no idea. But there's a pretty figure. And there's a pretty figure. Okay. All right. Isozymes actually are important, but they have nothing to do with what we're talking about. So why your book puts it in there, I don't know. I guess you're thinking of protein structure or something. All right. Well, there are other mechanisms for controlling things allosterically. So you've seen some shape changes, R and T, that have existed as a result of the binding of either CTP or ATP or aspartate locking the enzyme into a given configuration. There are other mechanisms that can involve allosterism that aren't quite the same way. And so I'm getting ready to show you one for a very important enzyme in our cells known as Protein Kinase A. Protein Kinase A, you're going to hear a lot about over the rest of this term because it's involved in a very, very important set of reactions called signaling. Okay? And I want to say just a word briefly about the phenomenon of signaling, and you're going to hear a lot more about it later. But in addition to cells being able to control their metabolic pathways, cells also have to be able to understand signals that come from outside the cell. This is essential for a multicellular organism. A multicellular organism has to know when the body is talking to it. Okay, that is, the individual cells have to know that the body is saying, hey, we got to do something. Very simple situation. I'm being chased by a grizzly bear. Okay? Grizzly bear is chasing me. I darn sure better get as much glucose into my bloodstream as I can so I can feed my muscles so I can get away from this thing. And that signal has to happen very quickly. And that signal has to be very effective at dumping glucose into the bloodstream. Well, that signaling happens through a molecule called epinephrine. You know it as adrenaline. And when adrenaline dumps into the bloodstream, the liver gets that message and goes, whoa, we got to start dumping glucose out real quickly. And the way that the liver cells do that is internal to them. They get a signal from on the outside that there's adrenaline there. And that adrenaline stimulates them on the inside to send messages to their metabolic pathways. Okay? The messages control the enzymes, and one of those ways of controlling the enzyme is with protein kinase A. So protein kinase A is playing a critical role in this process of signaling. Now, I will give an entire lecture or two in just a bit on the phenomenon of signaling, so I don't expect you to have all the things of signaling figured out from what I've just said. But suffice it to say that signaling is a communication that's happening at the molecular level inside of cells. The signal is coming from outside the cell, and the cell is responding to it accordingly. Okay? Protein kinase A is one of the things helping to communicate that signal. All right. Well, that's a kind of a mouthful, and I haven't even shown you protein kinase A on the screen. What I show you on the screen is another molecule that's very important in this process. It's called cyclic AMP. Cyclic AMP is one of the molecules that actually is the signal. Okay? So a signal comes from outside the cell. The cell starts making cyclic AMP because of the signal from outside the cell. And cyclic AMP is related to the nucleotide AMP. The cyclic part is, instead of having this phosphate just dangling up here in the air, it actually makes a ring, a second ring here, and it makes that little circle. That little circle right there is what makes it cyclic AMP and what makes cyclic AMP different from AMP. Okay? So cyclic AMP and AMP are very different things. AMP, as we will see in the cell, is a good indicator of the fact the cell is in very low energy. Cyclic AMP is an indicator of the fact that the cell is getting a signal. Okay, well, the reason I show you cyclic AMP is cyclic AMP communicates its signal by binding to protein kinase A. Protein kinase A looks like this. So protein kinase A is a multi-subunit protein. It has two catalytic subunits, and it has two regulatory subunits. So in that sense, it's a little bit like ATCase, and then it has multiple subunits, some of which act as catalytic subunits, and some of which act as regulatory subunits. But it's different in the way that the allosterism actually affects it. Okay? Now, what we see in the normal situation where there's no cyclic AMP present in the cell is the fact that the regulatory subunit is the regulatory subunits are associated both with each other and with the catalytic subunits. So in the absence of cyclic AMP, we have the situation shown here on the left, where the regulatory subunits are in association, and they're also in association with the catalytic subunits. The association of the regulatory subunit with the catalytic subunit blocks access to the active site of the catalytic subunit. So when we're in the situation that we have on the left, protein kinase A is inactive. It's unable to catalyze a reaction. When cyclic AMP is being made in the cell and the concentration starts to increase, cyclic AMP binds to the regulatory subunits, as you can see here. That causes, as you might suspect, a shape change, a very small shape change, and that shape change causes it to release from the catalytic subunits. The catalytic subunits now have their active sites completely open, and they're able to catalyze the reactions that protein kinase A catalyzes. Now, protein kinase A does not catalyze metabolic reactions. Okay? So not all enzymes catalyze the formation of metabolites. Protein kinase A, as its name suggests, okay, by the way, it doesn't suggest until I tell you what the things mean. A kinase, so you hear the name kinase there. Kinase is an enzyme that puts a phosphate onto something. Whenever you hear the term kinase, that's what it means. This enzyme puts a phosphate onto something. Well, protein kinase A means that the enzyme is putting a phosphate onto another protein. It's phosphorylating it. Now, if you remember from your basic amino acid structure that we talked about before, There are 3 amino acids that have hydroxyl groups on them: serine, threonine, and tyrosine. And protein kinase A can put phosphates onto threonine and serine. And you might wonder, well, why does it put phosphates onto threonine and serine? The reason it's putting phosphates onto those proteins is that the proteins it's putting it onto are themselves enzymes. Now, if I take an enzyme and it's sitting here in solution and I put a phosphate on it that's negatively charged, do you suppose I'm going to change the shape of the enzyme? I might very well. And if I change the shape of the enzyme, will I change the activity of the enzyme? And the answer is yes, I might very well. So we can see that through protein kinase A, we can affect, by covalent modification, and that's what it's doing here, we can affect by covalent modification the changing activity of an enzyme. You might say, well, does it turn it on or does it turn it off? And the answer is it depends on the enzyme. Some enzymes get turned on by phosphorylation. Some enzymes get turned off by phosphorylation. And we'll see that the cell has an incredibly beautiful symmetry with respect to phosphorylation and the turning on and turning off of different enzymes. Okay? So what you're seeing as a result of action of protein kinase A is that protein kinase A is, in fact, controlling entire metabolic pathways. It can control several metabolic pathways at once by phosphorylating a few enzymes in different pathways. Now, I'll just give you a brief heads up. We'll talk about it later in the term. But a prime example of this is the pathway that involves the breakdown of glycogen. Glycogen is a polysaccharide that we have in our muscles and our liver. It's a storage form of glucose. Breakdown of glycogen makes more glucose available to the body. Okay? Phosphorylation of the enzymes in glycogen metabolism, you don't need to know this, I'll talk about it later, but it activates the enzymes that break down glycogen, and it inactivates the enzymes that make glycogen. Same enzyme, same control. One mechanism of control, opposite effects on pathways that do opposite things. Yes, sir?
Is tyrosine not commonly involved in enzymes?
Tyrosine is commonly found in enzymes, yes. Tyrosine is not nearly as commonly phosphorylated in enzymes as these two are. And for a long time, it wasn't even realized that it was phosphorylated. But when people got investigating it, they found that not only was it phosphorylated in some enzymes, but the enzymes in which it was phosphorylated were really interesting enzymes. I'll talk about this more when I talk about signaling, but since you've asked, I'll answer your question. The enzymes in which tyrosine gets phosphorylated are commonly intimately involved with determining whether the cells divide or not. And what we see is that these enzymes are frequently what we call oncogenes, meaning that they are involved in causing a cell to develop uncontrollable growth. And so the development of that uncontrollable growth usually arises as some aberration in that control mechanism involving that phosphorylation. But I'll talk about that. Okay? All right. So there is protein kinase A. This is an example. Okay, if you look at this, you think about this. This is 100% effective. This enzyme, when it's bound to this regulatory subunit, it's 100% off. ATCase, we couldn't turn it off. But if we had every one of these guys bound like this enzyme would be 100% turned off because we've completely blocked the active site. This enzyme can't do anything. Okay. Yeah, we'll leave that alone. That's not a biggie. Okay, so that protein kinase A, which actually, it's interesting, protein kinase A is controlled allosterically, but protein kinase A modulates covalent modification. In other words, it causes covalent modification, but it itself is allosterically regulated. Okay. Well, There are other examples of acetylation that's involved in things, I'm sorry, not acetylation, but covalent modification that's involved in the cell. Again, your book is a little odd in picking up on acetylating lysine. It is, in fact, a covalent modification that happens to the amino acid lysine when it's in certain proteins. The most common place we see acetylation of lysine, however, is not in enzymes, so it's kind of odd to put it in enzymes at this point. But in fact, it's in our histones. Histones are the proteins of our chromosomes that wrap DNA. And if you think about lysine and what you remember about lysine, lysine is a positively charged, has a positively charged R group at physiological pH. Okay? And histones, as a result of being positively charged, associate very nicely with DNA, which is negatively charged. And so it's because of this interaction, they can wrap DNA up into what we see in the microscope as chromosomes. And that wrapping is very effective. But that wrapping is also problematic when we want to do things like make RNA or we want to replicate the genome, because that involves separating strands and getting enzymes in there to make RNA, to make DNA. And this chromatin, these histones, are basically blocking access to that. So one of the ways that cells allow that access is by acetylating histones. And by acetylating histones, what happens is the positive charge gets covered up. And if this is bound to a DNA molecule, it doesn't end up being bound nearly as strongly as it was before. There's less positive charge, and so it kind of loosens up. It allows other things to come in and access the DNA. So it's not an enzyme, but it is a protein whose covalent modification is very important in the cell. The more common types of modifications, and there are other modifications as well. I don't want to leave you the impression these are the only ones. We saw in the last exam, we saw a covalent modification for lysine and for, I can't pull it up, proline, okay, that existed in collagen. That's a hydroxylation reaction. The more common reactions that we see in cells, though, are in fact phosphorylations. So phosphorylations of side chains of serine, threonine, and tyrosine are not uncommon, and they're not uncommonly used to control these proteins. Some proteins get many phosphates put onto them, and it's not completely understood in every case how that pattern of phosphorylation affects protein's activity. Okay? So what if we phosphorylate 12 of the 15 possible sites that are in an enzyme? How does that affect it if we do it compared to, say, 8? And those answers aren't completely clear at this point. But all this figure is showing you is what the kinase is catalyzing. It's catalyzing the transfer of phosphate from ATP. So here's the triphosphate. The phosphate gets transferred over here onto the protein. There's the side chain of the protein there that it's getting transferred to. And we're left behind with ADP. And as a result of this, the protein gains a phosphate. Protein kinases are not uncommon at all in the cell. Protein kinase A is one we'll talk about a lot here. And we'll also talk about another one called protein kinase C later on. Oh, didn't mean to do that. We'll come back to that, I suppose. I'm not sure why that's there. Okay. So this figure is designed to show you just some very simple examples of different kinases that are out there and the kinds of things that affect them. So, for example, you've already seen cyclic AMP affected protein kinase A. Protein kinase C, we'll talk about later, is allosterically regulated partly by diacylglycerol. It's a component of membranes. And so forth. So there are various signaling molecules that affect enzymes that do important things in this signaling scheme. And again, we'll talk about that when I talk about signaling. Okay. Cells, as I said, are control freaks. And a control freak has to basically be able to manage all components of its existence. I'm sure you know people who are control freaks, and they probably drive you nuts. Cells are the ultimate control freaks. They are that entity unto themselves, and they have to be able to manage their own environment as much as they can. So whenever cells have a mechanism for, let's say, turning something on, If the cell doesn't have a mechanism for turning something off, it's in trouble. So when we turn on phosphorylation, okay, if we left everything in the phosphorylated state, the cell would have problems. I gave you an example of glycogen metabolism. I said that phosphorylation activated the enzymes that turned on the breakdown of glycogen. If those phosphates never came off, our cells would break down their glycogen, they would never make any more. So cells have to be able to take off phosphates once they get put on. And the way that cells take off phosphates is by using enzymes known as phosphatases. So phosphatases act counter to a kinase. Phosphatase acts counter to a kinase in that it removes the phosphate that a kinase puts on. Removes a phosphate that a kinase puts on. People ask, what's orthophosphate? You can see it right there. It's just simply a free phosphate floating around in the cell. Okay. It's a hydrolysis reaction. You can see the involvement of water. And you can see the product of that takes us back to the amino acid side chain that had the hydroxyl group and the free phosphate. So we've returned everything back to its original state. Phosphatases are important in glycogen metabolism, for example, in reversing the breakdown process. So if putting phosphates on activates breakdown and stops synthesis, then taking phosphates off has the opposite effect. It activates synthesis and it stops breakdown. That's very important because we want to be able to build our glycogen stores when we have plenty of energy. Our liver needs to have that glycogen stored away so that when we do get chased by that grizzly bear or we do have to run away from that thing that's scaring us, that we have that reserve to do it. So we have to have both situations where we can make and break down glycogen. Okay. What do I want to say here? Okay. So this may make a little bit more sense after I talk about energy later, but let me just introduce the topic here. What this figure is trying to show us is different levels of energy that we have relative to a protein with no phosphate on it and a protein with phosphate on it. That's the most important thing at the moment. Putting that phosphate on takes energy. It takes ATP energy in order to do that. And I told you that virtually all reactions that occur in cells are reversible. Virtually all reactions are reversible. So why do we even need a phosphatase to take off the phosphate? Don't we just have to simply reverse the reaction in order to take that phosphate off? Why can't the kinase take the phosphate off? Well, reactions are governed by concentrations of things. If ATP concentration is high, there's no way we're going to take that phosphate off with a kinase. And that's part of what this is showing us here, is that this reaction is strongly favored in the forward direction, and getting that phosphate off is going to be problematic if we're relying simply on concentration. So as a consequence of that, cells have evolved phosphatases, so they don't have to rely on concentration to get that thing off. Instead, the phosphatase will target this and remove it, and because the energy of this guy is higher than the energy of this guy, all right, that phosphate readily comes off, and the cell is able to exert the control that it does. Is it the most efficient process? Well, energetically, it's not the most efficient process, but it's the most effective process because it keeps the cell very much in control of its enzymes when it needs to. So the cell sacrifices some energy in the interest of control. The ultimate in control freak is exactly what that is. Okay, so you see that the topic of control here is that of covalent modification of enzymes. All right? And the covalent modification of enzymes involves more than just adding a phosphate or taking a phosphate off. And yes, taking a phosphate off is also a covalent modification. They're both covalent modifications, the addition and the removal of that. But there's another interesting mechanism that's used to control enzymes that is important in the body, and it's that of breaking peptide bonds in an enzyme. Breaking peptide bonds in an enzyme. Okay. Well, let's see. Let's talk about this here. So, one of the important, I shouldn't say one of, but a very important process in our body is that of digestion. Okay? We eat food, and our body magically breaks that food down and makes those nutrients available to us, whether that is in the form of energy, like it is for sugar or fat, or if it's amino acids, if it's in the case of proteins. And that can also be a source of energy, ultimately, if necessary. The digestion process means that our digestive system has to have enzymes that allow us to break down those individual components. And we've talked about proteases already. So proteases, of course, help us to break down proteins ultimately into substituent amino acids. And those amino acids can be used by our body to make other proteins as we see fit. I didn't say anything to start about those proteases themselves, and now I want to spend some time thinking about them and pose a problem for you with them. There are many proteases that our body makes that are actually synthesized in the pancreas and released into the digestive system. Okay? That's all fine and dandy. And the problem that arises is if I take a protease and I put it into a tube, what did I say happened if I let it go long enough? It ate itself up, because it'll start breaking down. That's all a protease does, is it starts breaking proteins down. Well, what if the pancreas makes these digestive enzymes, and the digestive enzymes start eating the proteins in the cell wall of the pancreas? What happens? Well, what happens is actually something that's a human condition that's known as pancreatitis. It's not a very good thing. It can be very painful. It can be fatal if it's not treated properly. And so what I'm planting in your head is the fact that these enzymes are not released by the pancreas in an active form. They are released in an inactive form. And then later in the digestive system, they are converted into an active form. The inactive form that they're made as are known as Zymogens. So an inactive form of an enzyme is a Zymogen. Z-Y-M-O-G-E-N. You can see it right there. Okay? Now, what Zymogens allow the body to do is just what I said, that an organ like the pancreas can release this in its inactive form. It doesn't attack the organ that's making it, for example. And then later in the body, in the place where it's needed, in fact, it can be converted into an active form. Now, there is, unfortunately, not a consistent naming scheme for these, but you can see sort of a pattern here. Here's pepsinogen, made actually in the stomach. Pepsinogen is an inactive form, and it gets activated to make the active enzyme known as pepsin. Chymotrypsinogen is the precursor in the inactive form of chymotrypsin that I've already talked about before. Trypsinogen goes into trypsin. But then we have other enzymes in the older nomenclature that have the word pro in front of them. The word pro also is used at one time to indicate a precursor of the active form, procarboxypeptidase, and proelastase, ultimately to make elastase. How do we get pancreatitis if we release things in the inactive form? Are we accidentally releasing things in the active form? And the answer is no, we're not. But what's happening is things are backing up to the point of the pancreas where things are being released. So if we have, it's actually in the digestive system that we see the active enzymes, I'll show you in a second, that start breaking these down. And if they start working backwards towards the pancreas, we can imagine we're going to be activating things that we don't want to activate. And as I said, it can be a very painful disorder. Anybody here ever have pancreatitis, or know anybody who's had pancreatitis? Do you? Yeah. Have you had it?
No, I haven't. I know somebody and I know lots of dogs.
A lot of, oh yeah, sure. Fatal for dogs?
Often.
Yeah.
Not often, but it is.
So how do you know a dog's got pancreatitis?
Well, there's a variety of enzymes that we look at. Basically, we look at the blood levels of,
So the dog looks like it's in pain.
and they have what's referred to as a Peptide, and then they take a prey position.
Ah.
Very painful.
Yeah.
So,
Yeah.
that's what my friend who had it said it was like too.
Yeah, yeah. It's of course in people, I guess people can talk and say, "Hey, wow, I'm hurting," and the owner may not recognize that for a while, which makes it more of a challenge for a dog. But definitely a debilitating and painful disease, and can be fatal. Okay. Well, so that leads us to our discussion then about the activation of these guys. So Zymogens have to be activated. And I'm going to show you an activation for one zymogen. And I'm not showing you this to get you to memorize a bunch of numbers and a bunch of different things here, but I'm really rather just showing you something about this protein so that you can learn something about what's involved in an activation and how it might work. I'll show you an enzyme structure in a second that will hopefully help to clarify that for you a little bit. Chymotrypsinogen is made as in an inactive form. It has 245 amino acids. Okay? And when it is synthesized, it passes ultimately into the digestive system, where normally it would be activated by trypsin. Trypsin would make a single cut in this molecule, and what it would do is it would break a peptide bond between amino acids 15 and 16. Okay? That creates an enzyme that is partially active that's known as pi-chymotrypsin. Okay? And pi-chymotrypsin has the ability to cleave other pi-chymotrypsins. So pi-chymotrypsin catalyzes basically self-cleavage, and that self-cleavage does a very minor thing. Look at this. It's cleaving off. It's cleaving between 13 and 14, and that causes 14 and 15 to disappear from this final structure. It's also cleaving over here between 146, 147, and between 148, 149, and that causes those two to disappear from the structure. And we're left ultimately with what we now describe as chymotrypsin. Chymotrypsin is completely active at this point, doesn't need any further activation, and will go and start digesting essentially anything. Okay? So, good exam question, all right? How do these guys stay together? Yeah. Well, hydrogen bond would be a good guess, but there's a better one. It takes a lot of energy to hold these together. They're in a very harsh environment. Disulfide bonds. Okay. So disulfide bonds are what's holding this guy together. So if I were to take pi-chymotrypsin and I were to if I were to take trypsin, if I were to take trypsinogen, I can't talk today. Breathe deep. Breathe deep. Okay. If I were to take chymotrypsinogen and do this cleavage before it folded properly, I wouldn't have any active chymotrypsin because the pieces would go flying away and I wouldn't have active enzyme. So again, the folding is necessary before we have this process happen, and that's a very good indication of it right there. If we look at the structure of chymotrypsinogen and we compare it to chymotrypsin, we see some very subtle changes that happen. One subtle change is up here. You see that this is a bond that gets broken, and this breakage of the bond now opens up a section of the enzyme, and it's opening of this section of the enzyme that actually allows access to the active site of the enzyme. All right? As a consequence, the enzyme becomes completely active once we've completely opened that up. And until we've opened that up, the enzyme can't do anything. It's not unlike we had the situation with Protein Kinase A, where we had that regulatory subunit physically blocking access to the active site. In this case, a portion of the chymotrypsinogen is blocking access to the active site, and this enzyme in that situation is 100% off. Okay? So it's very effective. There's other slight changes that happen. You can see very slight movements that happen in this protein as a result of these cleavages as well. Well, how does this process all happen? This is one schematic. There are other schematics showing the activation that go even further than what this one does. But this shows one set of activations for a bunch of zymogens that are important in digestion. So in this figure, the master enzyme for doing this conversion of the zymogens to the active form is enteropeptidase. Enteropeptidase, you see, converts trypsinogen to trypsin, and trypsin is one of the major players in activating all of these other enzymes in this process, as you can see. Okay? Not all of these enzymes are proteases, actually. Okay? This is a protease, chymotrypsin is, elastase is a protease, carboxypeptidase is a protease, but lipase is not. Lipase is an enzyme that breaks down fat. And so, again, controlling where these enzymes are active is important, and this is accomplished through the use of Zymogens. Okay, now, I'll tell you a fun story. Not a fun story, but at least a medically relevant story. I find a lot of people like those type of stories. There is, cells are control freaks. Cells have ways of turning processes on. They want to have ways of turning processes off. And so trypsin is a pretty powerful enzyme. And so being able to control trypsin is important. And so cells have an inhibitor of trypsin called trypsin inhibitor that is important in that process. And trypsin inhibitor, kind of like the regulatory subunits of ATCase, inserts into the active site and basically blocks access to it and prevents the trypsin from acting. So one of the ways of controlling trypsin is by having this inhibitor present that can later be removed, et cetera, et cetera. And when people discovered this enzyme, this inhibitor, they called it alpha-1 antitrypsin. Alpha-1 antitrypsin turns out to be important not just for regulating the activity of trypsin, but also for regulating the activity of the enzyme elastase. Okay? Well, elastase is a protease, and elastase is an important protease that's found in various places in our bodies, and one of the places that elastase is found is in our lungs. Okay? Why do we have proteases in our lungs? Well, we are breathing a lot of things in there, maybe being able to break some of those things down that we have in our lungs, and there's other reasons to break things down in our lungs. Elastase is important in our lungs for being able to break things down. Well, how does the inhibitor figure into that? As I said, cells need to be able to control levels of protease, because one of the things that happens is if we don't properly control the level of elastase that we have in our lungs, elastase attacks lung tissue, not unlike we see in pancreatitis, and causes damage to the lung tissue. Overactive elastase is actually implicated in emphysema. Okay? So elastase it really, really needs to be controlled, and it turns out that alpha-1 antitrypsin is a very important player in that control process. We have alpha-1 antitrypsin in our lungs to help us regulate the level of elastase that we have. Okay? Well, there's more to the story than that, because alpha-1 antitrypsin has an important methionine residue inside of it. Methionine has the structure that looks like this, as you can see. And the sulfur in this molecule can be oxidized. It takes reactive oxygen species in order to oxidize that methionine. But it turns out that this methionine is right at the place where it binds to the active site of elastase. If this methionine gets oxidized, as you see on the screen, it can't bind to elastase. Elastase stays active, and you get emphysema. Do you guys see where this is headed? Guess how you oxidize methionine? Guess where the reactive oxygen species come from? Smoking. So smokers, okay, have much more damage to their alpha-1 antitrypsin. As a consequence, they have more active elastase present, and as a consequence of that, they're much more likely to develop emphysema than a non-smoker. Okay. How much time do I have? Okay, I've got a little bit of time. All right, so we see a lot now of this regulation of enzyme activity, and regulation and control is very important. The last thing I'm going to talk about today is a very important zymogen system that we have in our body, and I think it's a fascinating one. And I'm only going to scratch the surface of it because it has a lot more depth than what we have time to go into here. But it's the phenomenon of blood clotting. And blood clotting is remarkable when we think about it. All right? Our blood is flowing through our veins and arteries at a fairly rapid rate. We don't have an enormous excess of blood. We need to have that blood flowing freely through our system when we don't have damage that's happening, for example, cutting an artery or something like that. Okay? But if we do have damage or a cut or a hole that's placed there, we need to be able to very rapidly plug that hole. Okay? So if you say, well, duh, we need to have blood clotting. Well, the process really almost has to happen with a hair trigger. Because if we don't have this clot forming fast enough, we can have sufficient blood loss that we die. Okay? So what that means, and because I say we have to have a hair trigger, is the blood has to have flowing through it the materials necessary to make the clot in relatively high concentrations. It has to have it present in relatively high concentrations, because if it doesn't have those things present in relatively high concentrations, it's not going to be able to form the clot fast enough and you're going to die. All right? Well, I'm talking about Zymogens, and Zymogens are a prime place to think about this. Is we don't want our blood clotting at random times. We don't want our blood clotting as we don't have some cut we have to stop the blood flow of. Okay? So we have to have these Zymogens present so they can be activated, and they can be activated fairly quickly. And they're activated in a scheme called a Cascade Scheme. All right? So I'm going to show you this and then step you through it. I'm not going to show you this whole scheme. I'll show you the scheme. I'm not going to take you through the whole scheme. And I'm not going to hold you responsible for it. But I want to describe what a Cascade Scheme is, because you're going to see a Cascade Scheme later when we talk about metabolic pathways. The Cascade Mountain Range is called the Cascade Mountain Range for a very important reason. Okay? We have cascading waterfalls that come down. And if you've ever done much hiking in the mountains, you know that the higher you go on the mountain, the smaller the streams are, because the streams coalesce, and as they coalesce, they get down to the bottom, they make big rivers, etc., and that's how we have the Willamette River and all the various other things that are out there. That big river is the result of cascading systems. Okay? This is a series of cascades, but the cascades are a little bit different than waterfalls, although I think the analogy is still appropriate. Okay? What we see are a series of proteins that are going from an inactive state to an active state to an inactive, and one protein activating the next one in the scheme, activating the next one in the scheme, not unlike I talked about metabolic pathways, one molecule being the precursor of another molecule, being the precursor of another molecule. But the difference between metabolic pathways and enzymes are that activation of one enzyme, when I make one enzyme active, that one enzyme can very rapidly go out and make another 10 or another 100 active. And those 10 or 100 can make another 10 or 100 or another 10 or 100. So we can see amplification of this signal that can happen very, very intensely and very, very rapidly. So cascading systems in the body have the effect of making a signal move very fast and having an enormous amount of amplification. Well, that turns out to be pretty important when we go to make a blood clot, because a blood clot is a polymer, and it's a polymer of a zymogen. It's not a chemical reaction, but it's a polymeric reaction. We'll see there's some chemistry involved as well, but we're making a polymer. We make the polymer from monomers. So we want to have a ton of these things ready to start polymerizing when necessary, and that's what's happening in this cascade. Okay? Well, I'm going to focus our attention on this process, on these proteins down here, prothrombin going to thrombin, fibrinogen going to fibrin, okay? And fibrin is the monomer that makes the clot. Fibrin is the monomer that makes the clot. Okay? So we see amplification, we see amplification, we see amplification. The next to last step, prothrombin is the inactive form. It's converted to thrombin. Thrombin, in turn, converts fibrinogen to fibrin, and fibrin forms the basis of the clot. Okay? It's important these guys be present in an inactive form, because we don't want these things going haywire before the clot is needed. Can we form clots in the absence of damage? We can, and they're very nasty when that happens. Okay? This is the structure of the monomer. I call it it's the monomer, but it's actually a dimer is what it is. It's the monomeric unit of the polymer, and it's fibrinogen. Fibrinogen has a structure that schematically looks like this, and we see a couple of relevant things here. We see these big guys here called B's, and we see these big guys down here called A's. And what they are are little tips at the end of each protein that get removed. And the removal of these causes fibrinogen to start to polymerize. So prothrombin is cleaving the big A's and the big B's off of this basic unit. Okay? All right. Now, if we take that, and once we remove those, I hope to show you here how it is that the polymer actually forms, or at least describe it to you. Okay, so here is the inactive precursor. Thrombin has cleaved, I may have said prothrombin, I meant thrombin. Thrombin has cleaved those A's and B's off of there. That leaves these ends open, and these guys are able to insert themselves into the appropriate unit. So the B's go into what was labeled the betas on the previous figure. We see the betas and the B's. When we have that big B on there, it won't fit in there, but we put the big B, we cleave the big B off, and now it can fit into there. And we start seeing, excuse me, the assembly of this guy into a lattice. Well, it turns out it's a 3D lattice, because what this is not showing you is that where the big A's were, they can fit into these gamma subunits here. And that gives a three-dimensionality to it. We can imagine this clot now is polymerizing both outwards towards us from the screen and in the plane of the screen as well. Well, that's really important, because a clot does have a three-dimensionality to it. A plot has to be very intricate, and a clot has to be able, ultimately, to stop water from flowing through it. Okay? It's watertight. Well, you get a scab. You know, of course, what a scab is like. It has a three-dimensional nature to it. And that scab is happening as a result of this polymerization that we see of fibrin. Okay? Very cool thing. Fibrin, in turn, during the process of forming a clot, there is what's formed initially, what's called a soft clot. That soft clot is kind of held together by various associations, including hydrogen bonds and all the various forces that we've seen before. But a clot needs to really hold itself in place. It needs to hold itself tight so that it doesn't let blood leak, etc. So a soft clot is turned into what's called a hard clot by action of an enzyme known as a transglutaminase. And what the transglutaminase does is it causes covalent bonds to form between glutamines and lysines, like what you see here. And those glutamines and lysines make a covalent bond, as here, and these are on separate subunits of that fibrin unit. We have just hardened the clot, because now it's being covalently held together, where before it was only being held together by relatively weak associations. Okay, so the clot has hardened. We have protected the cell against, not the cell, but the body against the loss of blood as a result of this. We have an important clot. All right. Well, I need to talk about a couple things relative to regulation of this, and then we'll call it a day. So, first of all, you hear about blood thinners. Okay? Blood thinners are called blood thinners because they inhibit the clotting process. All right? The clotting process, it turns out, requires a modification of prothrombin. Now, prothrombin, you remember, was the precursor of thrombin, and thrombin was the enzyme that converted fibrinogen into fibrin. The modification of prothrombin requires the addition of carboxyl groups to the side chains of glutamates. Okay, I'm going to come back to this figure. I jumped to this figure too soon. Let me come back to this. Okay. All right. So, what is happening is that the modification of prothrombin causes the glutamate side chains in prothrombin to gain an extra carboxyl group. So the original glutamate had a side chain that looked like what you see in black with one carboxyl group. The modification has added a second carboxyl group. Now, the addition of this second carboxyl group is very important, as we shall see in a second. Okay? Everybody see the reaction that's happened? So the side chains of prothrombin have this extra carboxyl group. Well, what does the addition of the extra carboxyl group do? What it does is it favors the binding of calcium by prothrombin. It favors the binding of calcium by prothrombin. If prothrombin only has a single carboxyl group, it can't bind to calcium. And it turns out calcium is present at the site of the wound. This is a way of getting prothrombin at the place where it's going to be needed. Notice we're still in the prothrombin state. We're not at thrombin yet. Okay? So in order to get prothrombin effectively held at the site of the wound, we have to have these carboxyl groups on there because the carboxyl groups bind to calcium, and calcium is present at the site of that wound. Okay. Now, this is where the blood thinners come in. Okay? Vitamin K is a necessary precursor used by the enzyme to put those carboxyl groups on. Vitamin K is necessary for putting the carboxyl groups onto prothrombin. Okay? Blood thinners, okay, mimic, they look enough like vitamin K that they block the enzyme from working with vitamin K, and they block the carboxylation of prothrombin. If prothrombin can't be held at the site of the wound, you're much less likely to make a clot. You're much more likely to bleed. Why would you ever want to have that? Well, if you are prone to making clots internal in your body, like maybe you've had stroke or you've had some other issue in the body and there are people who have various conditions that lead to that. I haven't, my step-grandmother actually has phlebitis, which is a very painful clotting problem in the legs. And so she takes blood thinner to reduce the likelihood and reduce the problems associated with clotting, so she's not having clotting accumulating where she doesn't, the prothrombin accumulating where she doesn't want, and doesn't have this problem happening. Two of the blood thinners that are commonly used you see on the screen. Warfarin, you might recognize if you read the boxes of rat poison. If you have nothing better to do, so go to the store and read the box of rat poison. You'll see that the active ingredient is warfarin. Warfarin works by thinning the blood of rats. Okay? And we use warfarin, in fact, in these blood thinning reagents that we give to people. And one of the things that you discover is when you give warfarin to a person or you give dicumarol to a person, that you have to be very careful how much you give them, because if you give them too much, what happens is they have virtually no ability to clot, and then they hemorrhage. You can die of hemorrhaging. So you have to be very careful. They're actually titrated, and people have to go in and get monitored very carefully for how much clotting factor and how much clotting actually is going on. And these work by inhibiting the action of vitamin K, putting that carboxyl group on. Okay. So let's see. We're almost there. We're almost there. Any questions about that? I'm kind of going through, not asking you guys if you have questions, comments. Everybody ready to get done? Okay. I've got a cool song that goes with this. I want to finish this last thing. We'll do the song. Okay. So the last thing I want to talk about is the removal of clots. Okay. Bodies, like cells, have to be able to not only make clots when necessary, but remove them. We don't get a scab and it stays on our body forever. We have to do something about that. Well, I've already talked about, my God, we're hardening this clot. We are putting some things together to keep that clot as stable as possible, but in fact we have to get rid of the clot to some extent. So there's a system for removing the clots as well, and the system for removing the clots involves an important protease called plasmon. Okay? Plasmon. Plasmon is synthesized, not surprisingly, as a zymogen as well. The zymogen is called plasminogen. And so we don't want to break those clots loose until we're ready to. Plasminogen is inactive. Plasminogen gets converted to plasmin by action of a protease that's very important from a health perspective. It's known as tissue plasminogen activator, or as you're more likely to call it, TPA. TPA has the fame of being the first recombinant protein that was ever approved for use in human beings. And it was approved because of the ability of tissue plasminogen activator to open up clotted arteries. Okay? This shows an artery in a heart that's plugged by a clot before treatment with TPA. After treatment with TPA, TPA activated plasmin, and plasmin dissolved the clot and opened this artery up. Okay? Now, TPA is as you can imagine, a pretty important protein. We don't use it to treat all clots. We don't use it to treat things routinely because it's a pretty powerful tool. We don't want to have hemorrhaging and other problems happening. But in places where people have had major heart attack or have had a major stroke, and there's a suspicion of the involvement of clotting in that the treatment of a person with TPA on a routine basis shortly after that can happen, can have major reduction in the effects of a stroke, for example. So if people who've had a stroke resulting from a clot are treated with TPA, many of the side effects of a stroke actually can be reduced if it's administered in the first couple or 3 hours after that happens. Okay. That's what I want to say. I think that's all for here. I do have a song if you'd like to listen to it. If you have to go, that's fine too, but I think you'll enjoy the song if you want to hang around. Let's see. Turn that on. And here it goes. We'll see if it plays. Oh, and I've got words for it too. Hold it. You can sing if you want. Okay, there you are. Now, you got to have it all down from that song, right? So, all right. See you guys on. We have a 4-day weekend.
Yeah.
weekend.
See you Tuesday. Amen. Yes, you too. Have fun. A lot of fireworks. Actually, you know what I'm doing on the 4th of July? I'm marrying somebody.
You're marrying?
I'm conducting a wedding ceremony.
Oh, wow.
And I wrote the ceremony because she's an old friend of our family, and she said, would I do it? And I said, yes. And I said, what do you want me to do? And she says, I want you to do something funny. So the pressure is on. If it's not funny, it's